Hello everyone. Today I am going to discuss about Hatch and Slack pathway or C4 cycle. In introduction part, I must say that a major new pathway for carbon flow during photosynthesis other than C3 cycle or Calvin cycle was first established in sugarcane, maize and other grasses rather be called as tropical grasses. And the plants which actually go through the C4 cycle are termed as C4 plants, and the first stable product of C4 cycle is 4 carbon containing dicarboxylic acid like oxaloacetate. And the C4 plants possess a unique type of leaf anatomy that is known as crunch anatomy. And if we go through the history, that is, if we focus on the discovery of C4 cycle, then we will see that initially Kaprilov in the year 1960 in Russia, on Giames and Kortchak, Hart and Burr in the year 1965 in Hawaii, reported that 4 carbon containing dicarboxylic acid, aspartate and malate were major early products of sugarcane leaves when the plants were exposed to 14 CO2 for just 1 sec. Later in the year 1966, Hatch and Slack in Australia confirmed the previous reports and revealed that a new cyclic reaction mechanism and this pathway is now referred as C4 pathway, and the plants which possess this pathway are termed as C4 plants. And now the unique features of C4 plants. C4 cycle has been found to be operated in many plant species of Poaceae such as maize, sorghum and few other plants also like Atriplex, Amaranthus, etc. And these plants can be distinguished by some typical or unique characteristic or rather be called as unique features. In these plants, the first stable product, photosynthetic product, are 4 carbon containing dicarboxylic acid like oxaloacetate, malate and aspartate, and there is complete absence of photorespiration, and they possess as earlier I said that a special type of leaf anatomical structure, that is crunch anatomy. Now what is crunch anatomy? Actually it is a German word and it means wreath or ring. In the leaves of C4 plants, the vascular bundles remain surrounded by bundle sheath of larger parenchyma cells, which in turn are surrounded by smaller mesophyll cells. Actually, the chloroplast in the bundle sheath cells are larger in size and lack grana, but the chloroplast in the mesophyll cells are smaller and contain grana. In maize, plasmodesmata have been observed which connect the bundle sheath cells with mesophyll cells. The word, as earlier I said, that crunch is a German word and which means wreath or ring, and the radially arranged bundle sheath cells around the vascular bundles look like a ring or wreath. That's why it is called as crunch anatomy. Now these are the reactions of C4 cycle. It involves 2 carboxylation reaction. Of them, one reaction occurs in the chloroplast of mesophyll cells and another in the chloroplast of bundle sheath cells. The primary carboxylation of C4 cycle is done by phosphoenol carboxylase. In this reaction, the carboxylation of phosphoenol pyruvic acid happens and produces 4 carbon containing oxaloacetic acid. It occurs in the chloroplast of mesophyll cell. And then the oxaloacetic acid immediately equilibrates with other C4 dicarboxylic acid, aspartic acid and malic acid. These reactions occur in the presence of transaminase and NADP plus specific malate dehydrogenase respectively. And then the malic acid gets transferred from mesophyll cells of chloroplast to the chloroplast of bundle sheath cell, and then it gets decarboxylated to form CO2 and pyruvic acid in the presence of NADP plus malic enzyme. After that, second carboxylation occurs in bundle sheath chloroplast. RUBP or ribulose bisphosphate accepts carbon dioxide in presence of RUBP carboxylate, that is Rubisco, and finally yields 3-phosphoglyceric acid as in case of Calvin cycle. The pyruvate is then transferred to mesophyll cells chloroplast where it gets phosphorylated into phosphoenol pyruvate, that is PEP. It is then catalyzed by pyruvate PI kinase. And Hatch and Slack pathway starts with carboxylation of phosphoenol pyruvate and not RUBP. It is because the former has great affinities with CO2 than the latter. In recent years, minor variations in the mechanism of C4 mode of photosynthesis, that is Hatch and Slack pathway, have been observed in different C4 plants. The main difference is in the way the 4-carbon dicarboxylic acid is decarboxylated in the bundle sheath cells, and on the basis of these, 3 categories of C4 plants and also of C4 pathway are recognized. The first category, that is there are some C4 plants such as Giamase Saccharum officinarum, Sorghum sudanense. They utilize NADP plus specific malic enzyme for their decarboxylation purpose, and the mechanism of C4 pathway is said to be of NADP plus Emi type. And the second category of plants such as Atriplex spongiosa, Portulaca oleracea, they utilize NAD plus specific malic enzyme for this purpose, and the mechanism of C4 pathway in these plants is said to be of NAD plus Emi type. And there are also a specific type or third category of C4 plants such as Panicum maximum, Chloris guyana, etc. They utilize PEP carboxykinase enzyme, and the mechanism of C4 pathway in such plants is known as PCK Emi type. The mechanism of C4 pathway described earlier is of NADP plus Emi type, but it shows generalized picture of the C4 pathway. That's why before going to different categories of C4 pathway, I am going to start with NAD plus Emi type, just because I have described NADP plus Emi type as a generalized version. In NAD plus Emi type pathway, the oxaloacetic acid in the cytoplasm of mesophyll cells get transaminated by the enzyme aspartate aminotransferase. Again, I am saying the enzyme's name is aspartate aminotransferase, and this enzyme is present in the cytoplasm, and it requires glutamic acid as the donor of amino group. And the aspartic acid then transferred to the mitochondria of bundle sheath cells, that is Kranz cells, and in the mitochondria of Kranz cells, the aspartic acid is converted back into the oxaloacetic acid by reversal of transamination reaction. And the oxaloacetic acid then reduced by NAD plus specific malate dehydrogenase into the malic acid. Again, I am saying that is the most important stage, that is oxaloacetic acid is then reduced by NAD plus specific malic dehydrogenase into malic acid. And this malic acid then decarboxylated by mitochondrial NAD plus specific malic enzyme into pyruvic acid and CO2. The CO2 then diffuses from mitochondria to chloroplast, where it is reduced to carbohydrate through Calvin cycle. The pyruvic acid diffuses from mitochondria into the cytoplasm, where it is transaminated to yield a specific type of amino acid, that is alanine, and the enzyme involved is alanine aminotransferase, and the glutamic acid acts as amino donor. And the alanine then diffuses from cytoplasm of bundle sheath cell, that is Kranz cells, to the cytoplasm of mesophyll cells, where it is converted back into the pyruvic acid by reverse of the actually the previous reaction. And the pyruvic acid now passes from cytoplasm of mesophyll cells to the chloroplast of mesophyll cells, where it is converted into the phosphoenol pyruvate or PEP, and they utilize 2 ATP molecules for their energy. This is the third type, that is PCK type or PEP carboxylase type, and in this pathway the reaction sequence is quite similar to that of NAD plus Emi type. The main difference relies in the oxaloacetic acid in the Kranz cells is directly decarboxylated in PCK type, and secondly mitochondria are not involved in PCK type. Let's focus on the significance of Hatch and Slack pathway. The significance of this pathway is not clearly understood till now, but it has been suggested that this pathway is a modification of Calvin cycle, and it is advantageous to the plants which grow in the tropical area where the carbon dioxide concentration may be very much reduced, and there has been a reduction of atmospheric carbon dioxide concentration since the evolution of photosynthesis, and these might have prompted C4 plants to select this pathway. And the discovery of this pathway has indicated that there is existence of yet undiscovered photosynthetic reaction other than the conventional C3 cycle or Calvin cycle. So these are all about C4 cycle. In this lecture session, I have discussed about the brief history of C4 cycle discovery, different types of or variation types of C4 cycle exhibiting plants, and the brief biochemical reactions of C4 cycle, and the significance of C4 cycle. So thank you all.